All right. There's actually 2 mini lectures. They're not as many as I would like. So we'll try to get through these without taking too much time, but also feel free to ask questions. Okay, so we're going to talk about alignment and the kind of high-level concept of alignment, some of the specifics of this HiSat splice-aware aligner that we're using, although it's going to be like a pretty brief treatment given how much there is to that algorithm, as you'll discover if you try to read the paper. And then also kind of dig into the outputs that it's going to create, like try and understand this BAM file format. So alignment is kind of important in bioinformatics. You could say that it's like central to most genomic research. This is a very typical genomics bioinformatics workflow where you're getting FASTQ data from your sequencing instruments or cores, and often the first step you do, no matter what your downstream steps are, is align to a genome, and then do some kind of quality control. And then in these green boxes is kind of like the more exploratory phase where you might find peaks or quantify transcripts like we're going to do, or find SVs or many other things. Yeah, there's a question. You don't see it? Oh, you're trying to follow along.
The other ones?
They're linked from the RNA bio, from the, I think these are linked from the first page of the alignment module, module 2, I think. They're also in the Google Drive, but I think I put them in a subfolder called mini lectures. You see? Yeah, so there's this.
I will put the link in the Slack.
Okay, yeah. Drive. Sorry, there's a lot of mini lectures, so it might be hard to find the right one. I'm sorry about that.
But I also think Nia put it on the GitHub page for CBW.
I put the module 2 big lecture, not the mini.
Oh, I see. Okay. Okay.
They're on like the intro slide, not the actual.
Oh,
Yeah.
well, there we go.
Or not intro slide, like the intro.
The model.
Yeah.
For RNA, on RNA bio.
Yeah.
Yeah. So there are multiple places, but I think Nia will put direct links in Slack just to make it super clear. Okay, yeah, so there's this kind of secondary phase of doing things like finding peaks and quantifying transcripts. And then there's kind of like this like endless iterative phase of kind of ad hoc analysis and interpretation and discovery and visualization. But it all starts with alignment. So how does alignment work? At like a really basic level, this is kind of about fitting pieces, which are the reads, into the correct part of the puzzle. And the reference that the Human Genome Project produced for us, if you're doing human analysis, is kind of like the picture on the box of the cover, right? So you're like looking at this reference and saying, I have this piece, and trying to figure out where it would best fit. And if you are able to find a place where it fits, but there's like a slight imperfection in how it fits, that can indicate something interesting, like maybe a variant. RNA-seq alignment comes with some unique challenges, some shared challenges and some unique challenges. It can be computationally expensive. There's a reason why we're downloading and aligning these kind of like unrealistically small data sets, because if we had like a realistic data set, it would take probably hours. Of course, there are introns, right? So we've talked about this. We're aligning something that's representative of a messenger RNA in most cases, which has been spliced, but we're aligning it back to the genome. So we have to be aware of introns. We have to be aware that we expect like large gaps in our alignments if the read happens to span an exon-exon junction. Ideally, we would be able to align our data with just one approach and be done with it. This is a little bit more true than it used to be. Like there is kind of standardization around alignment and alignment approaches, but it's still the case that depending on what your downstream application is, you may need to like tweak your alignment strategy a little bit or use a different aligner. So certainly most people are not using the same aligner for their DNA data and their RNA data. And then even for RNA data, they may be like doing a couple of different alignment strategies depending on their end goals. Like for example, if you're hoping to detect fusions, you might run your aligner in a little bit different way that supports that application. There are different kind of very high-level alignment strategies for RNA-seq, and they're kind of broken down in this old paper into 3. We're going to be primarily focusing on this aligned to the reference genome approach, but there are certain situations where you may want to align directly to the transcriptome, and there may be other situations where you don't have a reference genome or transcriptome, or you don't want to be limited by them in some way, and you attempt just like a de novo assembly of the transcripts in your RNA-seq data. So which of those strategies should we use? It kind of depends. So for de novo assembly, you might do that if your reference genome doesn't exist. So I think at least one person in the room mentioned having potentially that problem. Or for cases where there's like really complicated regions of the genome, where there's like a lot of polymorphisms or mutations, things like the HLA region in humans, they're just highly polymorphic, that where it's really not possible to have a single reference genome that like works for all people. And when you try to align one person's sequence data against another person's reference, the alignments don't work well because people are just very different in those regions. And so that can sometimes cause you to want to do like a more of a de novo approach. But there's other solutions to that problem as well. You may want to align to the transcriptome if, for example, you have short reads. So this is less common now, but when you had like really short reads, you often didn't have enough sequence where you had a good alignment in one exon and another part of the read in the next exon, so that you could correctly identify both places in the reference. So when you had like really short reads, especially like 30 base pair reads or something, it was pretty hopeless to do like a splice-aware alignment to the reference. And so you might try and align directly to the transcriptome. But in most other cases, by far, I would say now the most common approach is to align to the reference genome in a kind of splice-aware manner that allows you to both use information about known transcripts, but also allow for discovery of novel isoforms. But there are pros and cons to these. This is kind of a fun old figure of just like how many different aligners there have been over the years. This is really stabilized now. I feel like there are a few like pretty dominant aligners. Like if you're doing DNA alignment, practically everyone is using like BWA-MEM. If you're doing RNA alignment, I would say most people are using HiSat or STAR. But there may be specific applications, especially for some of these other kinds of omics, where you may use a different aligner. Should you use a splice-aware or unspliced mapper? I think from like the various hints along the way, you've obviously probably grokked that we are using a splice-aware aligner. Generally, that's a good idea because the fragments we're sequencing are from mRNA with the introns removed, and we're lining back to the reference genome. So you want to be aware of splicing. So again, unless your reads are really short, a splice-aware aligner like HiSat or STAR is probably going to give you the best results. And so we're going to use HiSat mostly just because it works really easily with the other kind of tool chain that we've chosen for this workshop, right? So we're using HiSat, string tie, and a little bit, what's it called? Ball gown. I was thinking like cummerbund, all these different tuxedo, ball gown, that will just work very well together because they're all developed by the same group from the same lab. But in our own like pipelines, we actually use, I think, STAR more commonly. So this is not like a strict, you must use HiSat, but that's what we're going to use. So we're going to just talk a little bit about how it works. this is just a peer behind the curtain a little bit, right? Like there's a whole field in science that's math and statistics that like makes these aligners possible, which we couldn't possibly cover here without like turning this into like an algorithms and math course. But just to give you like kind of like a high level idea of how it's working. It's using the Burroughs-Wheeler transform and FM indexes. And it has this kind of hierarchical strategy of using multiple types of indexes for alignment. So it does a kind of genome-wide index, and then it has numerous, actually 64,000 local indexes, where it's taken the whole genome and broken it into smaller pieces and built kind of more refined indexes for each of those pieces. And it's going to use these two indexes to align data both accurately and quickly to the genome. And it's also going to attempt to use information about known variation and known transcripts to do this alignment in a very smart way. So as I said, it uses this hierarchical indexing strategy as well as some adaptive strategies that we'll talk through with some cartoon depictions. So the general strategy is to first find like the general location in the whole genome with this global index. So we're going to map a small part of each read until we find a kind of match globally across the whole genome. And then we're going to pivot to these local alignments to really figure out the specifics and complete the alignment of that read. And this is done like primarily for performance reasons.
It's like a word find.
Yeah, it's like a word find. That's exactly right. Yeah. So it's going to do the paired reads separately, but if it really has trouble with a read, it will attempt to use the mate to kind of give it a clue of where to start looking. So we're going to use this cartoon to kind of walk through some scenarios to kind of illustrate what HiSET is doing. So imagine that you have, if I can use a pointer here, yeah. Imagine you have this like very, very simple gene with like 2 exons and a small intron in between, right? So this is much simpler than the typical case, but just for illustration purposes. And then you have 3 of these orange reads that fall into the transcript from this simple gene, right? So here's the transcript where exon 1 and exon 2 are connected and the intron has been removed. And now you've got 3 reads from this transcript, one of which is like just inside one of the exons, and the other 2 are overlapping the exon-exon boundaries to different degrees. So how would HiSET approach these alignments? So it does this global search, right, using the global index. That's what's represented with the blue read. And it's basically doing like someone in the crowd said, a word search. Like it's taking a short piece of the read and it's looking for exact matches somewhere in the reference genome. And it's looking for a minimum of 28 base pair perfect match to one or a few locations in the reference genome. And as soon as it finds those hits, like it finds somewhere in the genome where part of the read aligns, it then switches to this extension mode. So now instead of like going to the next part of the read and taking a chunk of it and searching across the index for a match across the whole genome, which is very time and computational intensive, it says, I found this small match. Maybe I'm in the right place of the genome. What if I just look to the like next to the match and ask if it's right? And if the basis match, I'll just keep extending. So I found like a kind of like a pointer, like maybe I'm in the right place. And if I extend the alignment, like, does it work? Basically, is it a perfect match? And as long as it's a perfect match, it just keeps extending. And in this case, because this read was fully included in this exon, once it found that little 28 base pair match, it was able to just extend the rest of the read. Like the rest of the read just matches here perfectly and it's done. This extension part is much faster than the index searching part, right? So that's like a space or a time-saving innovation. So that's the simplest situation. What about these more complicated situations? So here you have, again, a read, but now it doesn't match perfectly within an exon. It's also going into the next exon. So here it does the same starting approach. It tries to find a chunk of the read, up at least 28 bases that matches in this global index search. Once it finds one or more of those matches, it does this extension step, right? And it's just like, okay, does this base match? Does this base match? Looking good, looking good, looking good. But then it gets here and it's like, the read doesn't match here anymore. So then it pivots. It made it 93 bases in this case. It pivots and says, these last 8 bases aren't matching here. Let's find where they do match. But now, instead of searching the whole genome, I'm just going to search the local index because I have a place in the genome right now. I know the read is like here somewhere, but it just doesn't match perfectly this one region. Maybe there's some splicing happening, but I don't need to look on like other chromosomes or like way down the chromosome because there's a pretty good chance that it's like within, I don't know, a few thousand or 10,000s of bases, right? Because introns aren't that big. I mean, they might be like up to 1 million bases in some cases, but they're not like on a different chromosome, obviously, and they're not on different, right, really different parts of the chromosome. So it uses this local index now to do a search, and it looks for a match of that 8 bases. And in this case, it finds a hit. It's like, okay, if I look over here, I find an 8 base pair match, and I'm done. And it's also, as you might have like observed when we built our index, we told it where the introns and exons are, right? So it also has some clues about like where it might start looking within that index. It can say like, first, let's assume maybe this is a known isoform and just go say here, I have an alignment that's to an exon that's making it to the end of the exon and it's mismatching. Let's look at some of the other exons over here that I know about and see if it matches there, right? So we can do that in this smart splice-aware way. And the final scenario is just the same thing, but even just a little bit more complicated, where it kind of iterates between these phases. It does the global search to find this minimum 28 base pair match. Then it's like, okay, I think I know a place in the genome that looks right. I'm going to do this extension step. Once the extension fails, I'm going to go to my local index and do a search. And it's like, aha, I found an 8 base pair match at this other exon. And then I'll do another extension step. And then I've completed the alignment. So it's kind of like global search, extension, local search, extension. And then it does some checks for compatibility. Like, does this alignment make sense? You know, it'll ask things like, oh, I'm sorry, like, does the alignment in this exon, is it in the same orientation as the alignment in this exon? You know, it wouldn't make sense for it to like be aligning one way and then flip to aligning the other direction, for example, if it's a proper splice alignment. Yeah, there's a question.
What if that exon-exon junction appeared in the global search region in that first 28 bases?
So the question is, what if the like exon-exon junction was like kind of disrupting that first 28 bases? Yeah, that's a good question. I forget exactly the details of how it handled that situation. It either like moves the 28 base pair starting point that it's looking for down, or yeah, I'll have to check to like see the correct answer, but it has like some logic to handle that situation. Yeah.
So, do you mind just clarifying what this like prefix size mean in this context? I mean, as I understand it, like if this is in this case is that first 8 base pairs was matching to multiple possible like subsequent exonic region. So yeah, so what happens after that?
So someone's asking about this prefix size point. So if the first 8 bases, this first 8 bases of the part that hasn't yet been aligned, where we're going to the local index, it could be that there's that first 8 bases of the second part of the read matches multiple places, even in the smaller index. That could definitely happen. And so what it's saying is it'll just increase the prefix size. So instead of looking for one exact match of 8 bases, if it finds multiple, it'll look for 9 base pair matches and 10 base pair matches until it finds a unique match in that local index. Another question?
Yeah, sorry, I'm not too sure. Like, if you know where the splice is in the transcript and then in the reference genome, could you not just search using that exon junction itself? Like, you just grab the smaller middle piece of the sequence to do like a more efficient search?
Yeah, so the question is, if you know where the exon-exon junctions are, couldn't you just like basically check all the known exon positions rather than like relying on this index? The answer is yes and no. So it depends on which like kind of mode you're running in, but you want it to be aware of those so that it can like quickly find those solutions if those are the most like parsimonious solution, if you will. But you don't want it to be limited to that because you want it to be able to make alignments that aren't in your known transcripts file. Because this is like not limiting us to only detecting known isoforms. It can find a splice alignment for a novel isoform that's not yet annotated. And that happens all the time, right? Partly because we don't totally understand all of the transcript isoforms of every gene. And also because sometimes like biology is just messy. Like if you look at basically any gene, it typically has more than one known isoform. And if you look at RNA-seq data, you will find that most of the data aligns to one of the known isoforms as you expect. Some of it aligns to what could plausibly be like a proper alternate isoform that just hasn't been annotated. And some of it aligns to just kind of like noisy transcription, where like to some extent the splicing machinery is not perfect, right? And it will often like at some low level splice together like kind of random stuff. And we don't always know whether that's kind of biological noise or real, but it's the truth as far as the sequence data is concerned, right? So like this is really trying to like find like what the true alignment is using the known annotations to guide it, but not limiting itself to that. And so that's the reason why it does like this local index and just doesn't totally rely on that known exon-exon boundaries.
Okay, so essentially it just avoids being too restrictive then.
Yeah,
Okay, perfect.
yeah,
Thank
it's
you.
avoiding being like restricting itself to just known exon boundaries.
So would it be possible to say like it doesn't match the exon region directly next to it? So would it just automatically switch to the next one and look for it like that?
Yeah, so the question is, would it like automatically sort of look at or be limited to like the next exon, and if it doesn't find something there, look at the next one? It's essentially yes. Like I don't know if it's like the logic of it is exactly that simplistic, right? Because it's the order of operations, it's doing this like local index search and it's using like nearby exons to guide it. But yeah, it's certainly not like limiting itself to just the next exon. It's like limiting itself to all possible subsequent exons or within some parameters. And there's all kinds of parameters you can set in HiSat that have to do with like how big introns can be, the kind of penalties for gap sizes and things like that that you can tweak under certain circumstances. Another question.
Yeah, does it always look for an absolute perfect match or would take into account any like mutations or?
Yeah, so the question is, does it always look for like a perfect match? I think it starts by looking for a perfect match, but it does allow mismatches, and those are also configurable settings. And you can also provide it with a file of known variation to kind of help it to recognize like, okay, there's a mismatch here, but that's a known polymorphism. That's not that concerning. But we'll see in the alignments that it's certainly able to like come up with alignments that have mismatches that either, represent base errors or variance. Yeah, and we'll look at an example. Yeah, another question.
And will be reporting like a unique match for the read. Will not be reporting an alignment on two genes because for some genes we may have some similar regions that could be aligned the same region.
Yeah.
Is it included or it will be just one unique alignment?
So the question is, will it report just one unique alignment, or can it report basically multiple matches? For example, in the case where you have like a highly related gene family where there are multiple kind of equally correct solutions. This is also probably a configurable parameter, but by default, it will allow multiple mapping. Yeah, that is possible that there could be like more than one solution. And those get recorded just in a different way in the alignment file. Like it will indicate, like I didn't find one good match, I found multiple matches. And then you or the downstream tools can decide what you want to do with those situations. that could be the right, like, it's the right answer in some cases, right? That like the sequence correctly aligns to both places, and it's just not there's not enough sequence difference between these two regions of the genome, and we can't differentiate between them. It's not wrong. It's not quite as useful as a totally unique match, but it could still be useful in some ways, right? You could like think about how you might, I don't know, average the counts across the reads, or you might want to use it in some way. So that's kind of like a downstream choice.
What we go to the account, say, is it gonna be assigned based on some logic for this case, or could be like assigning the count for the two genes?
Yeah, so the question is like downstream steps when we start trying to like count reads that support different genes or different isoforms, like what will it do with that information? We're going to see. It depends on the tool. It depends how you run it. Yeah. Lots of good questions, guys. Okay. I think we have like hopefully a general understanding of how HiSat works. You can read the paper. It's quite good. It goes through a lot. Like they do a very nice, thorough job of explaining. There's a lot to it. What do we get out of HiSat at the end? We get a SAM-BAM file. A SAM file is a sequence alignment map format file, and BAM is just the compressed version of that. So you can think of like BAM and SAM as the same thing. It's just that BAM files are compressed. It's kind of like our FASTQ file and our fastq.gz file, right? They're the same information inside, but just in a different format. But the compressed files require special handling. You can't just read them. You need tools that kind of understand the compression, which most tools do. So we're going to switch to this other lecture file to just quickly review something and dig into BAM files, SAM-BAM files. Hoping that this is still displaying correctly. So as I said, the BAM is just a compressed version of the SAM file. BAM files are usually indexed, so we're going to create BAI or BAM index files. You'll often see those right next to your BAM files. Just like the other applications of indexing we've seen so far, it kind of provides for a quick lookup or fast retrieval of alignments, so that if you're opening a BAM file in, say, a program like IGV, and you tell it to go to a certain position, it can like go there very quickly and see all of the alignments that belong there because this index file is kind of making that very efficient. We're going to talk about sorting. You often need to sort your BAM file in different ways to support different kinds of indexing or different kinds of analysis strategy. What does a SAM-BAM file format look like? This is a little bit scary at first. So if you were to like just read some of your SAM file, this is what it would look like. It has two main sections. It has a header section that has kind of global information about the alignments, and then it has an alignment section where each row, here there's some wrapping happening because we're just looking at this in the terminal, but each row has like 10 pieces of information about the alignment. And we're going to break this down into pieces. So in the header section, it's a good idea to kind of get used to this idea that like when you receive data from someone, sequence data, a fair amount of time, it could be already aligned data, right? So someone may be like, you could have the FASTQ files, but I've already aligned the data, or I'm making the aligned data available. And that in some ways is like a great time saver for you, right? It's like, I don't have to like waste time in compute cycles aligning this data. But you probably need to know how it was aligned and think about that for a minute before you just like put it into your pipeline and go on to the next steps. And a lot of useful information is in this header section. So you can use a tool that just like prints out the header of a SAM or BAM file, and there will be a header line that tells you information about like the format, the version, and the sort order. Like, is this position sorted, for example? There'll be a section called the reference sequence dictionary that tells you basically like what were all the sequences that I was aligning to. So the most common thing you might see listed here would be the chromosomes, right? So it'll list like chromosome 1 to 22 and XY that just says like whoever aligned this file, they aligned it to these against these contigs, against these things like chromosomes. But there can often be other stuff in there, right? So from that you can learn like did they include like a virus genome in the alignment because they were interested in like viral integration as well as human genome alignments? Did they include alternate contigs for the HLA region because they want to get better alignments of HLA? All kinds of stuff like that could potentially be in there. Then there's read group information. So this will tell you something about the sample. Like it might have information about like where it's a lot of this information comes from like the FASTQ read names, right? It's like preserving it in the BAM file. Like what instrument was it sequenced on, what sequencing center, and then what are information about the sample? Like is there a sample name or a library name? And then finally, there's the program information. So it might tell you like which aligner was run and which version, which could be really important, right? Like, was this run with STAR or HiSat, or was it run with a DNA aligner? And actually you want to rerun it, knowing that with an RNA aligner. So that's the header section. This is just another example, kind of like looking at that of what it could look like. So, here's like telling you this is version 1.3 and it was coordinate sorted, and there's like sequence names. So this is chromosome 20 and it's this long, and other information about the read group. The alignment section looks like this. So again, this is just kind of like printing it out to the screen. Some of it is like pretty immediately intelligible. Like over here, you can see your sequence read, right? So this should be like a direct copy basically from the sequence read in your FASTQ into your BAM file. And there's even something called an unaligned BAM file, which really is basically like a BAM version of your FASTQ file where it just has like the sequence read and the quality string and not much else. But this is an aligned BAM because it's got all this other information. So you've got your sequence, your quality string, and then a bunch of other stuff. Your read name, you probably recognize that, but a bunch of other stuff that might seem a little bit unintelligible until you dig into it. So in order, it has, I guess, 11 things. It has the query template name. So this is like your read name. It has this flag, which we're going to dig into. It has the reference sequence name. So this would be like what it aligned to, like chromosome 22, for example. And then where it aligned. So where's the beginning of the alignment? What is the quality, the mapping quality? So like how well did this read map? Someone was asking before about multi-mapping. So if it maps to like 100 places in the genome, it would have like a really low mapping quality score, maybe 0. A cigar string, we're going to talk about what this means. And then the reference sequence name and position of the mate. So it tells you where the read maps and where its pair maps. The length of the template. So this is kind of like giving you a sense, like if you have paired sequence data, what's the size, the inferred size of the fragment that was sequenced? And then the actual sequence and the quality string at the end. Any questions about these components of the BAM alignment section other than about flag and cigar, which we're going to talk about? Yeah.
So you just take the P next, and then you subtract the POS, so the position of like the one base leftmost, just to get the inferred template length?
Something like that, yes. Yeah, conceptually, yeah. I have to think about that, but yeah, I think that might be right, because it's like the start of read 1 and the start of read 2, and it's like the distance between them. But you could do that math and see if it's matching up, but it's something like that, yeah. Someone was asking basically if the T-len, the length, is just the position of the read versus the position of the mate subtracted, and I think it is. Okay, so this is again just showing you like here's one row of an alignment record from a BAM file, and we talked about most of these elements, but we kind of left for the moment the flag and the cigar string. So what are those? So flags are this like really useful, a little bit hard to wrap your head around, very efficient way of representing a whole bunch of information in one number. So the flag is a number, depending on the spec. Here it's a number between 1 and 2048. Or sorry, no, between 0 and 4095. So this number is basically describing the status of 12 binary states, 12 questions. So things like, does the template have multiple segments or not? Is the segment unmapped or not? Is there a secondary alignment or not? Is it a duplicate or not? So it's just like asking these status questions, and it's representing them in a kind of binary format, which is like a series of zeros or ones. So it's like 0, no, 1, yes, for these 12 different questions. And then it's taking all the possibilities of 0 and 1 and assigning a single number to them and putting that number in the file. And then there's kind of like a lookup table somewhere where it's like, okay, if I give you the number 1332, it can go and look and say, okay, that corresponds to 000101001 or whatever, which then tells you the answer to these 12 questions or 11 questions or whatever the number is. So that you could very like efficiently store the status of all of these things in one number. And it's mostly about saving space, right? Like rather than having that alignment section have 12 extra columns that are like yes, no, and think about all of the like the file size that would add, it's like compressing it down to this one number. Does anyone have questions about that idea? We're gonna like look at examples of it and like pull out reads that correspond to different flags and use a tool online to convert between the flag number and the status of all these questions. And I think by doing that, it becomes more clear. But are there questions right now about how flags work? Okay. The other question mark is this cigar string. So this is also another efficiency thing. It's kind of like when you do a blast alignment, right? Most people probably at some point have like taken a sequence and blasted it, and you get back that nice kind of like ASCII picture of like how your read aligns to the reference sequence, and there's like the little lines that tell you these bases match, and there's like a gap or an X or something or a star, I forget, where there's mismatches, and it kind of like really shows you like how this thing aligned against that thing, right? It's either like a perfect match, or it's like a match and a gap and a match, or a match and a mismatch and a match. But it's not very efficient, right? Like if you imagine like to take those kind of like blast representations and stick them in the SAM file, how big the SAM file would need to be to represent all that. So someone came up with this cigar string as a way of like really efficiently describing the same thing with as few characters as possible, so this file would stay as small as possible. And it does it with this concept of operations. So the cigar string is basically a sequence of lengths and operations to tell you which bases align to the reference or don't, which bases are either aligned or deleted or inserted or potentially represent introns. So you can do something like this, where you say 81M, 859N, 19M is a very like efficient way of saying there's 100 base pair read, which has 81 bases of match, 859 bases of gap, and another 19 bases of match. So can you kind of like visualize like maybe in a blast result, what that might look like, and then think about how this is just like a very, not very human readable, but barely human readable representation of that. Mostly it's read by computers, right? So like the downstream software can like take that and know, and like say an IGV show you, okay, it matched whatever, 81 bases here, and then there's this gap, and then it matches over here. There's like efficient way of saying how does the read align to the reference. Any questions about cigar strings? Okay. All right, so then just one note about cram files. You may have heard of these. This is actually like kind of the new standard. It's just an even more efficient compression approach. So one thing you can do if you know what your reference genome is, you can do some like tricks to basically make your BAM file even smaller. And you can get as much as 30 to 60% savings in the size by converting it from BAM to CRAM. And it does that by doing these sort of like diffs from the reference genome. So where it can say like, if these 100 bases just like match perfectly the reference genome, I don't need to like waste a lot of space in the file describing what the sequence is. I can just say, go to the reference and look it up if you want it, right? The reference has this sequence. And I'm just gonna tell you like, it matches the reference here. And if you need the sequence, go get it. So you're basically like pairing the reference genome with the alignments and saving a lot of space by kind of converting some of those alignments to kind of just like lookups, where you say like, instead of every row having to like have these sequences and probably in many cases like repeat the sequence over and over, even if it's just like a simple sequence that like is exactly the same as what's in the reference. And there's some other space saving conventions like, which are optional, like compressing the quality scores. So instead of having like every possible quality score from 0 to 40, you can kind of bin them and say like, I'm only going to store 6 possible quality scores. And kind of like say, if you're between 2 and 9, we'll just call it 6. And if you're between 10 to 19, we'll just call it 15. And by like having less quality string options there, it makes the compression more efficient. Any questions about CRAM files? Yeah.
Do you mind clarifying what like diffs refers?
Sorry, yeah, diff is, someone's asking what diff means. I guess that's like maybe like a bit of a computational term. So a diff is just like short for difference, where you can use something like for example, if you're writing code, this is a common application of the term diff. If I write some code and I commit it to GitHub, and then I make a little change, and I want to be able to see what the change is, I ask for a diff. I say git diff these files or whatever, and it'll say like, well, you used to have this, now you have this, and the difference is that you inserted this line here. That's all I mean by diff. Trying to think of an analogy. It's like word track changes, basically, right? Like if you've ever used Word and turned on track changes and you make a different, like an edit, it kind of shows you what the old version is and it shows you where the difference is. It's like that. So it's like doing a diff against the reference. It's saying like everything's the same as the reference except here, as like as a space saving trick. So one of the gotchas, like CRAMs, it all sounds good, right? You've saved a lot of space, but one of the gotchas, something you have to worry about is you need that reference now, right? And we've learned that there are different versions of references out there. And one of the things that can sometimes happen is someone like shares a CRAM file and you don't know or can't find the right reference to go with it, which basically makes the CRAM file useless or problematic at best. That's the trick with CRAM files, is like you need to make them in such a way that you're very clearly indicating, like in the header records, what the reference file is and making that available somewhere or keeping it handy. We've had problems where like we download data from dbGaP and we're not able to like correctly reproduce the BAM file from the CRAM file because we can't find the right reference. So just be aware of that. Sorting, we often sort BAM files by position, but you sometimes sort them by read name, and there's different reasons for that. Sorting by position is often done for performance. Specifically, building indexes usually requires that you first sort by position. You might sort by read name for other reasons, like especially if you want to like really be able to get at the two reads, like read 1 and read 2. If you sort by name, they're right next to each other. Sometimes that's convenient or efficient, especially in things like fusion detection, where You're really interested in like what happened to read 1 and what happened to read 2. And are they like separated by some unexpected distance or position or something? You've already had a brief introduction to bed files maybe, but just to mention them, these are often used together with BAM files. It's just a very common format to specify regions of the genome. It's kind of like an even simpler thing than a GTF file. They're usually in a format that looks something like this. There's all these variations of them, but the core thing is the bed3, the first 3 columns. It's literally just the chromosome, the start, and the stop, right? So you can use this to represent arbitrarily anything in your genome. Where are the exons? Where are some enhancers? Where was the exome target capture designed against, et cetera, et cetera, et cetera. So you can often use bed files to like represent a subset of the genome and then use that to pull out reads from your BAM file that correspond to that subset. And there are tools that we're going to use to manipulate all these kinds of files like SAM tools and Picard and bed tools. Some common sources of confusion. So these have come up already, some of them. So we'll just quickly talk through genomic coordinate systems. There are the sort of 0-based versus 1-based issue that we're going to talk about. There's different genome builds, right? So if you're familiar with human, you know that like sometimes you're working with HG19, sometimes you're working with HG20. There's complications with variant representation. So we actually talked through this and I said there was a slide coming. So this is the slide, which just graphically depicts what we talked about yesterday, that you have these 2 competing ways of thinking about the genome coordinates. One, where you're numbering the bases directly, right? So imagine you're at the very beginning of chromosome 1 and pretend that it doesn't have like an unsequenced telomere at this moment, but you actually know what the bases are right from the first base. There's a TA, C, G, T, C, A. And we would just call that first base number 1 and so on. In the 0-base system, we kind of number the positions between the bases. And as we talked about before, there's like some kind of computational and mathematical benefits to doing that. But the result of having these 2 different systems is that you have different representations. So like if I'm trying to indicate a single nucleotide, this say G here, I could say it's chromosome 1 starting at position 4 and ending at position 4, or it's chromosome 1 starting at position 3, ending at position 4, depending on which coordinate system I'm using. So you just need to be aware of these differences, and especially because, like, as I said, the bioinformatics field is like almost perfectly split between the 2. So GFF, SAM, VCF, Ensemble tend to be more 1-based. BED, BAM, UCSC browser tend to be more 0-based. And I'd say there's no one doing bioinformatics that isn't using like one from each of these categories, at least one tool or resource from each of these categories. So just be aware, like if you're trying to intersect something with something and you have this 1-base versus 0-base, you may need to think about the implications of that and do some kind of conversion. We already talked about this. I think this is pretty straightforward. There are different genome builds, right? Like coming up with a reference genome is an ongoing process. There are versions to it. There are different reasons why you might include additional contigs, like alternate contigs or HLA sequences or what have you. So this is just like a really common source of confusion and problems if you have any kind of mismatch between your reference genomes. You know, if you align against GRC38 and then try and visualize them in GRC37, nothing will make sense. And then the last thing, we're not really focused on variants much in this course, but just to be aware that even more so than like genome builds and coordinate systems, there's a lot of different ways you can represent a variant. And so this is an example where you have the reference sequence that's got like these 3 CAs, right? So there's this like slightly repetitive element, CA, CA, CA. And then you have an alternate version that's missing one of those CAs. And this can totally happen, right? This could be a somatic deletion. This could be a germline polymorphism. how to represent that. So all of these four or five colors are different ways of representing the same thing, right? You could choose to talk about the first CA being deleted or the last CA or the middle CA. You could like have a trailing or a leading base to say like it's the like let's look at this example maybe. It's like the GCA that's going to G to tell you that it's the first CA that is being dropped. So this is all just to say there are different ways of doing this. There are certain practices like alignment and parsimony that are recommended, but the field hasn't agreed on it or is not applying it consistently. So it's very possible that if you do anything with variance and you get a variant result from one person or group or tool and a variant result from another, and they don't match perfectly, that this is the reason. And so just be aware of that there's different ways of representing variance, and we haven't agreed on like one single way across tools and resources. Any question about that or about coordinate systems or builds? Yeah.
The variance, is there a method that you prefer? If we're looking at non-coding RNAs, with these modifications in them that introduce a large degree of mutations and all the variability, so downstream, like, is there something that you would recommend in that context?
What would I recommend? I mean, I will say that like the purpose of this kind of mini lecture that I think this is actually even in Wikipedia, this topic. Like one recommendation is to have like to certainly favor one kind of alignment, either left or right. Unfortunately, we can't agree on which should be the right one. And be as parsimonious as possible, meaning like have the shortest representation, like this is better than say this, where you're like giving more context than is needed, just for space saving reasons. And then like having, you know, one of these like reference bases for context is useful. So I guess like if there is a general recommendation, it's probably this bottom one. And there are tools that you can run to like convert your data to this, but yeah, it's kind of a Wild West out there. You had a question.
Okay. Going on what you're saying there, speaking to the human genome, if you start working with other alternative species that have poorly annotated genomes, and you know, like with dogs, we don't have any sequencing data that our facility isn't polyadenylated, just because there's no real way to do ribosomal depletion. Is it even worth looking at this? Like, is it worth putting in the time, knowing that there's a very limited amount of information that we're starting with relative to the human, which already isn't perfect?
So the kind of big picture question is if you're working with a non-human species, like let's say dog, where the resources and even reagents are more limited, so that like, for example, that you gave an example, you don't even have like good ribodepletion kits. So all of your RNA-seq data is like poly-A capture or poly-A enriched. Is it kind of even worth dealing with some of this stuff? Do you mean like, is it worth attempting variant calling, or
is it worth at all going beyond just doing differential gene expression and, you know, gene oncology and stuff like that? Like, is it worth getting into variants, isoforms?
Yeah, so is it worth going beyond kind of the basics of gene expression estimation and pathway analysis into things like variant calling and what was the other example used? Like isoform, isoform delineation. It's more, so I've actually done a fair bit of dog genome analysis, and it is more painful for the reasons you state. I think it's still, many things are still possible. It's just it's a little more work. The results you have to like treat with a little more caution. Like when you're doing, like when we take pipelines we've developed for humans and we adapt them, which we have done for mouse, for cat, for dog, we find that like the downstream results, even alignment is a little more tricky, but especially everything beyond that is just more noisy. You tend to have more false positives, but you can still you can still find cool stuff, and maybe you just need to do a little extra work to validate it, but
Is there a dog data set or reference genome that you recommend? Because there really isn't a widely established.
yeah, so the question is there a dog data set or reference that I would recommend?
Yeah, like I've used like the CanFam, I think it's last CFAM's the other one.
This might be like too specific a question for a long discussion here. I have a lot of thoughts about this.
We'll come find out.
Yeah, there are like 12 modern builds for dog, something like that. Like a lot of people have been like, we should improve the dog reference. And like they're fighting over which one should be kind of adopted. There's like several that are kind of leading the pack right now, pun intended. So yeah, I could like share with you what I've learned, but there isn't like a simple answer. And I've been bugging Ensemble for years to like fix this problem without getting much help. So yeah, we still use mostly CanFam, even though the CanFam 3 or whatever is kind of the latest popular stable release, even though it's vastly inferior to some of the builds that have come out since, because some of those downstream steps haven't happened. Like Ensemble hasn't made like a proper release of annotations for some of the better reference genomes.
Yeah, that was good. Right.
Yeah.
Top level files for anything else.
Exactly. Or there's no VEP cache, or like there's something you need in your pipeline that's missing. Yeah, I share your frustration. That's just how it is when you're working with like model or companion animal systems. You're always having to find workarounds. Another question?
Just quick question in terms of like prioritizing which, yeah, like which in terms of like which sorts of aligning strategy, like or aligning or like variants like classify strategy here. That does it suggest that like what's it so parsimony would take priority over like alignment? Sorry, I just sorry, I mean.
There,
It seems like there is like, not, okay, it's not necessarily about priority, but, yeah, so for example, say the penultimate like Haskell format where the position is as far left as possible, so that's a left alignment, but it's because it's longer, the variant is represented in a longer string that results in this case, if the bottom one is indeed the one taken, does that suggest that like parsimony is taking like priority over?
so the question is with this depiction I'm showing here of kind of different variant representations, is parsimony taking priority over alignment? I think that what this slide is trying to convey is that you should do both, that you should left align and trim to have the most, both like an aligned and most parsimonious representation as possible, with the caveat that you should include, I forget how they worded exactly, but basically you want to have like, yeah, reduce to as few nucleotides as possible without reducing the length of any allele to 0, which is why you're having that leading G, right? So like you would say that like CA nothing is shorter and more space consaving than GCAG, but the recommendation is like to not go all the way down to 0, to have that one reference base, because that is sometimes useful. But the GGCA to GG is like inefficient.
Right, even though it's like technically more aligned to the position is like technically.
Well, it's equally left aligned with respect to the variant. It's just not as trimmed as the bottom solution.
Okay.
So they're both left aligned, but one is not as parsimonious as the other.
Sure.
Any other questions? Edmonton?
No questions.
Okay. So I want to do a check on the schedule, or have we blasted through any breaks we were meant to have or anything? No, we're just continuing. Okay, so what we should do is just be done with lectures.